With that, we have two great students presenting today. The first is Sean Mahaffey. He is a master's student at UH Manoa and a National Science Foundation graduate research fellow. He studies under Dr. Brian Glazer in the Marine Geology and Geochemistry Division of UHM Department of Oceanography. His studies have focused on the interactions between terrestrial and marine realms, particularly the effects of land use on nearshore water quality and ecosystems. His thesis work investigates nutrient trends and anomalies in the streams of the Ala Wai Watershed. Sean will be defending his thesis this summer. And then next, we'll have Jessica Bullington, a master's student at the University of Hawai at Manoa, studying microbial ecology with Dr. Craig Nelson at the UHM Center for Microbial Oceanography. She completed her thesis on the environmental controls of a pathogen in the Ala Wai Canal with funding from Hawaii Sea Grant. Jessica was also the 2020 recipient of the J. Wadamel Merit Scholarship and the Graduate Student Organization's Diversity Merit Award. She has presented her research at Hanauma Bay, the 2019 Tester Memorial Life Sciences Symposium, and the 2020 Ocean Sciences Meeting. And without further ado, I'll have Sean begin his presentation. Thank you.
Excellent. Thanks, Michael, for the introduction, and thanks for having us. And thanks for everyone for tuning in. So today, Jessica and I will be co-presenting our talk, the Ala Wai Watershed, from nutrients to microbes. So I'm starting off with this map from 1887, just to highlight some of the changes that have occurred in the past 130 years. So if we zoom in on the Waikīkī area here, what you see is our fish ponds that were present at the time. And Waikīkī is naturally more of a marshland than what we see today. So if we jump ahead to 1920, we have a map with a proposed canal as highlighted here in the yellow dashed line in the Waikīkī area. Initially, it was supposed to have a second outlet into the ocean that never came to pass. And as you can see, the fish ponds that were there were filled in. There's heavily urbanization starting to creep in around the Waikīkī area. So a few years later, they started building the canal and subsequently started channelizing and altering the stream systems in the watershed. So if we jump ahead to present day and we enter SMART Ala Wai. So SMART stands for the Strategic Monitoring and Resilience Training in the Ala Wai Watershed. Now, the SMART project is very wide scope. It involves researchers from various disciplines and interests, as well as including or providing avenues for research for graduate students, undergraduate students, and local high school students to conduct research in the backyard of UH Manoa. So going through the talk today, I'll be doing part one, where I'll discuss the nutrient trends in the streams. I'll go over the site description, breaking down the different streams, some of the spatial patterns that we're seeing. I'll get to a couple of temporal patterns, and then I'll talk about the future work that I'm going to be doing. Now, this is all preliminary at this point for myself, so it's kind of broad stroke, and but we'll work from there. And afterwards, part two, Jessica will take over, and she'll be talking about the downstream effects of nutrients on microbial growth. She'll discuss environmental and seasonality of the canal, as well as her particular research and the sampling methods and the different patterns that she's seeing, as well as some predictive models and implications for future climate change. So let's reorient ourselves. We're on the main Hawaiian islands here on the island of Oahu. And if we zoom in over the Waikīkī area, I'm going to reorient the map slightly. So here we are looking from above. We have Waikīkī on the bottom. And just to get like a sense of where we are, I've included a couple features on this map. We have the Ko'olau Ridgeline on the top of the map, Waikīkī on the bottom On the left, we have Pu'awaina or Punch Bowl, and on the right, Leahi or Diamond Head. And in the center, we have the University of Hawaii at Manoa. So if we look, we've constrained this map into the actual watershed that we're investigating, shown here in the dashed yellow lines, and the 3 stream systems that we're looking at that contribute into the Ala Wai Canal there at the bottom. So the goals of my research are to describe the nutrient patterns in the watershed streams, spatially and temporally, and then also quantify the stream nutrient contribution to the Ala Wai Canal. These are our stream sampling sites, and I'll break these down, but as you can see, they have some kind of pattern to the numbering. Makiki is K, so the upland site in Makiki is K1 and K2 down at King Street. And then if we go over to Manoa, in the upper watershed, we have M1A, M2A, and M1B. These are different tributaries, A and B, that are merging into one another. And then it flows down to M3, M4. M5C is actually tapping off of the main stream and going to Kanibai Lo'i, which is a taro field near UH Manoa. And M5C is actually measuring the water after it goes through the taro patch, but before it returns to the Manoa stream. And if we go over to Pālolo, we have the 2 tributaries in the upper watershed labeled here, or the sites P1 and P1B will capture those signals, and they'll merge down to P2, which will then merge into Manoa, to the Manoa Pālolo discharge canal, and from there into the Ala Wai Canal. Now, there are some particular landscape features that we should also take note. In the upper Manoa watershed, we have Lion Arboretum, as shown here with the yellow star. It's in between M1A and M2A. So M2A will capture any kind of nutrient signal that changes throughout the Lion Arboretum. Also, we have Kanibai Lo'i that I mentioned at M5C. And in Pālolo, we have heavy agriculture in the upper Pālolo watershed, shown here with the green star, as well as there's increased channelization, and that's very like concrete channelized. There's portions of the stream that are just basically just shallow water flowing over concrete, and that captures any kind of signals we see at P2. Now, we've talked about the 12 stations that we used for sampling. We sampled over, we made 10 sampling days over the time period from July 2018 to March of 2019, approximately monthly. And there were several parameters measured. Today, I'll talk about a handful of them: temperature, conductivity, dissolved organic nitrogen, nitrate and nitrite, ammonium, total phosphorus, phosphate, and silicate. So we have the map on the left here that I've been using so far, but for the remainder of the talk, I'm going to refer to this simplified diagram. It basically represents the three river systems, Makiki, Manoa, and Pālolo, as well as the Ala Wai Canal, and also has the watersheds, or sorry, the sampling sites on them as well. And generally, we expect that all parameters, all nutrient signals will increase as we go downstream towards the canal. So moving on to looking at some of the data here, what I have shown here is 3 subplots that they're broken down by river systems. So you have Makiki, Manoa, and Pālolo. On the x-axis is distance from canal. So on the right side of each subplot would be the canal, and then moving to the left would be distance away from the canal, so going upstream. And then on the y-axis is the parameter of interest. Each site is represented by a different box and whisker plot, and they're connected via a straight line, which is the stream, if you will, flowing from the medians downstream. And the dotted lines or the dashed lines are the tributaries that are merging into the main stream. So if we look at temperature, we have this general trend. Specifically, if you look at the center plot in Manoa, you have kind of cooler waters in the upper watershed that's slightly warm as we go downstream. None of this is surprising. But the big standout here is in Pālolo at P2, we have increased temperatures. And again, this is at that heavily channelized and concrete area. So it's just absorbing a lot of solar energy. The water is very shallow there, and it's just heating up from that. If we move on to conductivity, in Manoa, in the center plot, we see a gentle rise in conductivity downslope. In Makiki, on the left, we have right off the bat just this heightened concentration of conductivity and with a slight increase downslope. And then in Pālolo, once again, at P2, we have this heightened signal. That is directly related to the warming temperatures. So as the water is evaporating, the ion concentrations are increasing, which is increasing the conductivity measurement. Moving on to dissolved organic nitrogen. We have a pretty stable signal throughout the watershed in Makiki and Manoa with some wiggle room from different sites. In Pālolo, we kind of have this heightened contribution from P1b. Now, this is the site that is downstream of kind of a increased agriculture area. So right off the bat, that's kind of putting, or it's suggesting that there might be some influence on the nutrient patterns in the streams. If we move on to nitrate and nitrite, again, similar signals in Makiki and Manoa. Manoa has this decreased value in the upper watershed that increases, but then is stable afterwards. But in Pālolo, we have this heightened value, once again, at P1b, again, downstream of the agriculture area. Staying on the nitrogen theme, we'll move over to ammonium. And this gives, Manoa has been fairly stable to this point, and this really changes things up. So we see this first increase at M2A. Now that is just downstream of Lion Arboretum. So it's, there's a few different processes going on up there, but it's definitely contributing to the increased ammonium concentrations. And we also have increased values at M5C, which is the return waters of the Kaniwai Lo'i. So also increased ammonium after the Lo'i. And we also have this increased value at P2, which is a bit perplexing right now. We don't have an answer for, but moving on to total phosphorus. Again, Makiki has this strange signal where it's just heightened from the get-go. It has some of the highest values throughout the entire watershed. And Manoa is fairly stable. We do have some little blips and wiggle room at M1b and again at M5c. But if we go over to Pālolo, we once again have this increased value at P1b downstream from the ag area. And now moving on to silicate. Makiki is fairly stable throughout its stream. Manoa has its pattern that it's been playing out for most of these parameters, where it has this slight increase at the headwaters and then fairly stable throughout. And then Pālolo just has very wide variability at its upper watershed or upper stream signals here. And in fact, P1b's range almost encompasses the entire range of the watershed for measured silicate. Now, just to summarize some of these, the sampling trends that we see, or the nutrient trends that we see is, in Makiki, we have high concentrations in conductivity and total phosphorus. In Manoa, we have increased ammonium concentrations after Lion Arboretum and Kaniwai Lo'i. And in Pālolo, we have high conductivity, dissolved organic nitrogen, nitrate, nitrite, total phosphorus, and silicate, all downstream of agricultural efforts up there in the upper watershed. And then in lower Pālolo, we have increased temperatures, conductivity, and ammonium. Now, moving on, I'm going to switch over to some color contour plots as shown here. And if we just look at the one on the left, temperature, I'll break this down for you. On the x-axis are the actual sampling dates that that occurred. And on the y-axis, it's broken down into separate streams again. So Makiki's on top, Mānoa, and then Pālolo. And then you have the different sites for each stream moving from upstream to downstream. So K1, K2 for Makiki, Mānoa M1A down to M6, and Pālolo M1A down again to M6, showing the convergence of Mānoa and Pālolo. And if we look at both of these, temperature and conductivity on the right here, we see a significant watershed-wide signal in January 26th of 2019. So really quick, I'm going to go to another plot of discharge. These are 5 discharge sites, stream discharge monitored by the USGS, and they are highlighted on the little plot on the bottom right with the yellow circles around those sites. So we could see an increased discharge in almost every location except for Makiki. But this green bar was the times that the samples were taken. So these are capturing the tail end of this increased discharge. So there was a rain event that was the initial cause of the discharge, and then likely that the remaining influx of discharge that it's capturing, just this tail end of it, is just this increased recharge of groundwater that's flowing through, and possibly continued rain a little bit. So we have heightened discharge. We'll go back to the temperature conductivity plots here, and that's why we see a decrease in temperature and a decrease in conductivity. It's just because we see this signal from increased water discharge. So we're going to compare 2 more parameters, dissolved organic nitrogen and silicate. And I'm probably reaching too far on this, and maybe I'm seeing something that's not there, and it's something that I need to investigate more. But if we look at November of 2018 to December of 2018, specifically in DON on the left side, dissolved organic nitrogen, we could see this increase of DON throughout the watershed. And now if we look at silicate on the right side, we also see a slight increase throughout most of the watershed. However, there is this kind of wrench in my thought process here. If you look at M1A, so the top, the headwaters of Mānoa, as well as the headwaters of Pālolo, M1A, or P1A, sorry, you have this decreased concentration, and then it increases downstream. So perhaps there's a relationship here, perhaps not. I need to investigate this more, and I'm going to look into the discharge between these 2 events, as well as doing some other statistical analysis with this. But real quickly, the watershed trends, temporal trends that we're seeing is a decrease in temperature and conductivity due to increased precipitation within a 24-hour period prior, as well as a possible relationship of DON and silicate decrease in November, December of 2018. So what's next for my research and my study? I need to investigate the trends that I'm seeing. I'm going to explore the geological history of this area, specifically in Makiki and the Tantalus Rift Zone. There's a relatively recent volcanism that has occurred. And that may play in part of why we have heightened values in conductivity and total phosphorus. I'll also look at anthropogenic factors and biological factors, both within the streams themselves and the surrounding areas. I'll also be jumping into stream discharge data and looking at this to see if it can help explain some of the trends that we're seeing, as well as creating a baseline or a base flow nutrient budget for the canal. all the sampling periods or all the sampling efforts that were done occurred at low flow conditions. So although we might be able to say something about a baseline nutrient contribution, it'll be hard to quantify an actual, like a complete budget, since we don't get these high flow events. The big takeaway is the streams have unique nutrient concentration patterns, likely due to Line Arboretum and Kanevai Lo'i in Mānoa, the upslope agriculture and heavy concrete channelization in Pālolo, as well as possible volcanic history of the Tantalus Rift in Makiki. Generally, nutrient concentrations do increase downstream from these and other processes, contributing significantly to the Ala Wai Canal. And with that, I'll leave you and I'll pass it on. As the rivers are flowing into the Ala Wai Canal, I'm going to pass this on to Jessica, and she's going to take it from here. Really quickly, I would just like to acknowledge my committee, Professors Brian Glazer, Margaret McManus, and Craig Nelson. I'd also like to thank sincerely Akabibi for his legwork doing the sampling, Jessica Bullington for her help with data acquisition, as well as the Mesh Lab, Stanley Leo, and Corey Yap for technical questions that I will always have. With that, thank you very much, and it's off to Jessica.
So I am Jessica Bullington. I'm another graduate student in the Department of Oceanography. And our understanding of the chemistry and discharge from the streams, as Sean presented, helps us model the population of a pathogenic bacterium, Vibrio vulnificus, that's living in the canal downstream. I'm going to start with a description of the Ala Wai Canal and then explain how we developed 3 predictive models of vulnificus. The Ala Wai Canal, which skirts Waikīkī, is approximately 4 kilometers in length and was constructed in the 1920s to drain the wetlands for development. It is cement lined and now dredged every few years to remove sediment buildup that deposits from the streams. And the major contributors are the Mānoa Pālolo drainage and the Makiki stream, which is here you can barely see because it's partially underground throughout the city. The canal is a partially mixed estuary, which means there's a gradient of salinity with fresh water at the surface from the stream inflow and salt water at the bottom from the ocean, and partial mixing of these 2 water bodies. This area has a mixed semi-diurnal tide, which means we have 2 highs and 2 lows in the day with different tidal amplitudes, usually less than half a meter impact on the water level. Another predominant feature is the flux of particles and nutrients from that stream inflow, and there's a significant spatial variation, which I'm going to simplify into sort of 3 major regions of the canal. The first is this stream inflow area, which has the largest gradient in salinity from the surface to the bottom. It's relatively cold water, turbid, with high nutrients. The second is the back end of the canal, which is not usually flushed by the stream flow from rain events or tidal flow. So it's this really stagnant water with high residence time. It's shallow, only about 1 meter in depth, turbid with high nutrients relatively. And we'll see later that it actually has the highest nutrient levels of all these regions. Our third and final region that we sampled is the area that would be affected by this, like a large rain event where the canal outflows into the ocean. So I'm calling it an outflow. It's usually sort of just an ocean region. It is deep, cold, clear with relatively low nutrients. And you can actually see the coral at the bottom, even in like 15 meters of water depth. So altogether, these 3 simplified regions of the canal each have really different characteristic biogeochemistry. But in addition to this spatial variation, we also have a big impact from seasonal changes to the weather. Thinking about seasonality in Oahu, we basically have 2 seasons, a warm, rainy winter and a hot, dry summer. In the winter, all the rain, which we are really experiencing this week, leads to increased stream discharge into the canal. And in the summer, the increase in air temperature warms the canal about 5 degrees Celsius. In our sampling efforts, we attempted to capture these two largest aspects of variation, the spatial and temporal variation. The SMART water quality sampling ran from October 2018 to September 2019 with samples collected approximately monthly. We hand-lowered sensors off of a boat to measure depth profiles of salinity, temperature, oxygen, turbidity, chlorophyll at all 18 sites, which I've labeled here. In addition, we collected bottle samples at 8 of these sites, here labeled in yellow, at 3 depths: the surface, the picnic line, which is the depth of maximum change in density, which is predominantly driven in this system by salinity, and the bottom water. These bottle samples were then analyzed for nutrient and bacteria concentrations. So I'm sure you want to know what patterns we're seeing with the nutrients and the bacteria. So we're looking at data of nutrient concentrations separated on the x-axis by region, and then the top and bottom panels by season. So the top being our rainy or our dry seasons, and bottom being our rainy season. And in this plot, total nitrogen is in green, silicate in orange, and phosphorus in red. And note that the scales are log transformed, and phosphorus is about 100 times lower than nitrogen and silicate. And just general patterns, we see that there are relatively high nutrients in the back end of the canal and relatively low in the outflow or ocean region. So we're seeing this accumulation of nutrients from the streams in this back end of the canal. In the rainy season, there are slightly lower levels of nutrients in the stream inflow. So how does this compare with microbial abundance? So now I'm showing in this right figure 3 additional measurements plotted in the same way. This time chlorophyll in green, which is approximating like photosynthetic microbe abundance, total prokaryotes, which are bacteria and archaea, in blue, and Vibrio vulnificus, the pathogen that we're going to focus on, in purple. And we see similar patterns as with nutrients. We see this high concentration in the back end and relatively low in the outflow. Therefore, nutrients might be a significant driver of microbial abundance and an unaccounted for driver of this pathogen's abundance. So now we're going to switch and focus entirely on Vibrio vulnificus. It's a bacterium that's related to Vibrio cholerae, the causative agent of cholera. When exposed, when you're exposed to vulnificus through an open wound or ingestion, it can cause a deadly bacterial infection. It is an estuary-specific organism, preferring salinity between 15 and 25 and temperature above 18 degrees Celsius. There are several reasons why monitoring vulnificus is particularly difficult. It requires expensive molecular methods to detect abundance. So measuring actually the Vibrio vulnificus hemolysin A gene, or VVHA, using quantitative PCR. Because it is naturally living in the estuary environment, we also can't use fecal indicator bacteria to predict its abundance, which we usually use for many other pathogens, since most of them are associated with sewage. And because of this expense and complication, the Department of Health is not continuously monitoring for Vibrio vulnificus, and it's relatively rare. So when we look at our data, we have about 15% additional variation in vulnificus can be explained when we include information about the nutrients, including nitrogen, silicate, and fluorescent dissolved organic matter, for a total model that can predict about 65% of Vibrio vulnificus variation. So my goal was to construct predictive models of Vibrio vulnificus based on our understanding of nutrients and water chemistry. And this has actually been done in the Chesapeake Bay on the East Coast, something similar. Let's see if we can get it to work here. So they predicted the probability of occurrence. So this is shown in the colors on this map. And we'd like to build something similar for the Ala Wai, but it's even better because it's using actual abundance and not probability of occurrence. So how are we going to do that? We have 3 predictive model frameworks that I'm going to share with you today. The first is based on predicting vulnificus from previous 5 days of rainfall and the previous day air temperature, which I did so that we could at least forecast 1 day into the future. And these measurements are in forecast of rainfall and temperature are made by the National Weather Service. This model gives us an overall daily average for the whole canal, but no spatial resolution. So you're getting one value for the whole canal and that outflow region. For spatial resolution, we need in situ water chemistry measurements. Our second model has those measurements for salinity, temperature, and dissolved organic matter, measured by fluorescence. This is our strongest model at predicting vulnificus of the 3 models that I'm presenting, but it's only at 1 location. Our third model gives us that desired spatial resolution, but it's based off of modeled water chemistry from a circulation model. And these measurements and forecasts for water chemistry for models 2 and 3 are maintained by the Pacific Islands Ocean Observing System. So we're going to go through each of these models and the predictions that we obtain and compare that for the sampling period that we have measurements for. Starting with model 1, which is based on seasonality, and we're looking at a time series that spans our sampling period. So it's October 2018 through September 2019. And we're looking at temperature, precipitation, discharge, and the predicted Vibrio vulnificus density. So this top panel in red is air temperature. Next down is blue in daily rainfall, which contributes to the stream discharge, which is green under rainfall, which is the volume of water flowing per second. And the rainfall and discharge, I've taken measurements from Mānoa Valley. And we use these measurements to predict vulnificus with this first model in purple in the bottom. So this is density of Vibrio vulnificus. So we're using that VVHA gene per milliliter, so you can think about it as cells per milliliter. And you can see that vulnificus abundance is really tracking these rain events in the winter, and overall sort of a baseline increase in the summer with warming temperature. So if we zoom in on a few months to kind of see that further, we have a snapshot of January and February in the winter. So you can see that it's warm but relatively colder than the summer, and particularly rainy. We have increased stream discharge and vulnificus increasing with these rain events. But in the summer, we don't have as much rain, but we have higher air temperature leading to higher water temperature, and we have this background increase in Vibrio vulnificus due to this higher temperature. And this model actually does a really excellent job of predicting the overall abundance throughout the canal when we compare it to our observations. And I should have pointed out that these dashed vertical lines are the sampling dates that we have measurements for. However, we don't have knowledge of what's happening in specific regions of the canal, just its overall average. For instance, this might be important if you wanted to know where you could go surfing that might be safe. If we use time series data from sensors maintained by the Pacific Islands Ocean Observing System at the Hawaii Yacht Club, this is our second model, which is in the Ala Wai Harbor. You can see the map marking the region or the one location in the top right. We can add additional information of water chemistry to help improve our predictions of vulnificus. And if we just, so we'll have same thing with temperature in red at the top. This time I have air temperature in pink and water temperature in this darker red. And you can see that the air temperature has a lot of fluctuation and the water temperature doesn't quite have as much fluctuation, but the overall pattern is the same with increasing temperature in the summer. Below that, we have salinity and precipitation, which are related. So in blue, the darker blue is salinity, and we have salinity decreasing significantly when we have these strong rain events, and you can see that in the lighter blue of precipitation. For about half this sampling time, we also have measurements of fluorescent dissolved organic matter, and this is in brown, in and then again predicted vibrio vulnificus density as the bottom in purple. This time I've log 10 transformed it so you can see more of this variation. And if we just isolate that January-February time again, we have higher amplitude fluctuations in vulnificus relative to just using the climate data. But it's only one location, albeit a useful one, since most of the recreational activity in this area is happening there. But what if we want to know if we can surf in Waikīkī? So this is our third and final predictive model. In order to capture spatial variation, we used a circulation model, the Waikīkī Regional Ocean Modeling System, which has hourly predictions at 40-meter resolution for 14 depth horizons. And on the right-hand side, we're looking at the surface 25-centimeter depth horizon for the model results. And I've isolated, again, that February rain event that we've been looking at in the time series data. And now we're looking at a map with modeled vibrio vulnificus concentration. It's log transformed, so the darkest red is going to be 1,000 gene copies per milliliter. And as we watch the rain event, we can see that the pulse of vulnificus reaches these high recreation areas of Ala Moana to the west and Waikīkī to the east. And I think this is pretty awesome that we were able to do this, but how do we use these different models? I'm happy to take suggestions actually on this, on model implementation. But for an overview, Model 1 is particularly useful because it has a 14-day forecast from the National Weather Service, and you don't even have to go down to the canal to make these predictions, but there's no spatial resolution. Model 2 uses robust measurements of water chemistry, but not really forecasted data. And Model 3 has the spatial resolution that we're after, and a 3-day forecast into the future, but it depends on modeled water chemistry. So my recommendation is to try to integrate these different frameworks in addition to deploying more sensors to measure water chemistry and integrate that with our spatial model, and then validate occasionally with measurements of vulnificus directly. One interesting implementation, given vulnificus's dependence on air temperature and rainfall, as well as this mixed salinity environment, is that we could use climate change predictions far into the future to assess the impact of climate change on this pathogen. So this is something that I'm trying to do right now, not fully analyzed, but we think about sea level rise and flooding, which I'm showing here on the right. This is about 1 meter sea level rise, which is projected to be what it's like at 2100 at the end of the century. And you can just see that there's potentially a lot more habitat in this area that has this mixed salinity water that vulnificus likes. In addition, we can use the dependence on air temperature and rainfall to kind of assess this overall abundance. So if we think temperature is going to be increased and there are going to be more frequent rainfall events, we'll have an overall increase in the abundance of this pathogen. And with hurricane flooding, we've actually seen historical outbreaks of vulnificus due to that type of flooding. So that might be an additional impact of climate change. Ideally, we can be prepared for the future and try to mitigate this situation that could be potentially really dangerous. So with that, I'm just going to summarize a little bit of both our talks and we'll be ready for questions. So from part one, which was Sean's presentation on the watershed nutrient dynamics, the streams have unique nutrient concentration patterns between Mānoa, Pālolo, and Makiki. Mānoa due to Lion Arboretum and the Kanawai Lo'i, and Pālolo upslope agriculture and heavy concrete channelization. Makiki possible volcanic history of the Tantalus Rift. And then we move downstream, impacts to the Ala Wai Canal, and the impacts on Vibrio vulnificus. So the canal is accumulating nutrients from the streams, and that's promoting microbial growth. If we include nutrient and dissolved organic matter measurements, that improves a lot of our predictive modeling of Vibrio vulnificus. But we have three particularly useful model frameworks based on measurements that are already being taken. And if we project that far into the future, we kind of have this concerning outlook with climate change. So we should do more research into this pathogen so that we're really prepared for the future. And I just wanted to say thank you to Hawaii Sea Grant, who funded this project, in addition to my committee members, Craig Nelson, Greg Stewart, Margaret McManus, and Anu Heimer, who have been contributing to our analysis, as well as Smart Ala Wai, including all the interns and volunteers, which it took a huge team to collect both the stream data and this canal data. And I wanted to thank the WRRC for inviting us to give this talk. And I believe we're going to have a little wrap up and then time for questions. Thanks.
Fantastic presentation, both Sean and Jessica. I really enjoyed all that you presented on, and we have a couple of questions, but I think if I'm going to skip ahead for 1 second. I wanted to ask you, Jessica, since my former career was in human health, are there particularly vulnerable populations for Vibrio that these forecasts could be used for, or would it be applicable to kind of the general public or folks using, like you said, for surfing or swimming?
Yeah, I think it's applicable to the general public, but primarily people that are using the area. And from what I've observed, most people that are actually sort of back in that canal area are paddling. So might be a concern for them. And they also report frequent staph infections. So we know that they're exposed to the water and they're reporting infections.
Thanks. So our first question comes from Sierra Payne. How did you verify your models? I'm not sure if that was directed at Sean or Jessica, but you both can answer.
Yeah, so I haven't extensively done the model validation, but I'm comparing them to our observations. So using, basically comparing our predicted abundance to our observations. And I've done this for our first model, and it does a really excellent job at matching up basically with our observations.
Thanks. Sean, did you have anything to add or?
No, I don't. Thanks.
Okay. Next one was from Catherine. How did you measure DOA?
I think maybe Sean can answer that. DON, right?
Yes.
Sure, yeah. So in the stream system, we did not directly measure DON, but total nitrogen was measured, as well as nitrate, nitrite, and ammonium. So it's kind of an indirect method. We just subtracted the inorganics from the total nitrogen to come up with the DON number.
Next one comes from Corey, it's directed towards Sean. Where were the water samples collected by an arboretum? And then also below/above the Lo'i in Manoa.
So I don't know the exact locations, as I did not do them myself. But in Lion Arboretum, so the two areas that were sampled were above the lower Lion Arboretum area, so above like the all the structures and the community center and things like that up there. And then below, I think, was just after all of that. So pretty close to the Arboretum. And then in the Lo'i in Manoa, in lower Manoa, I believe the water samples were collected right as the water was being tapped off into the Lo'i from Manoa Stream. And again, I don't have the exact location for the where it was sampled after, but I'm fairly certain it was after the Lo'i, but before the water was returned into Manoa Stream.
I'm going to go to Corey. Also asked a follow-up question. Can you speculate why the high levels of ammonium at the study site next to St. Louis School? Were the samples taken on channelized or unchannelized portion adjacent to the school?
That's a great question about the speculation. I'm not exactly sure. I'm not certain. And again, unfortunately, I don't have the information right now of exactly where those samples were taken, whether they were on the channelized or unchannelized portion next to St. Louis School. Sorry, Corey.
Next one, Chris asks, what is the relative load contribution from non-point stormwater source from Waikīkī in the super urbanized area?
So I'm guessing, Chris, I don't know if you're asking like, what is the load contribution into the canal or from the canal out into the nearshore waters?
Chris, feel free to unmute yourself and.
Yes, like you have the three streams contributing to the Ala Wai. But I would suspect in that urbanized area that the non-point sources from Waikīkī are a big load contribution, especially on big storm events, that plug flow. So I just wanted to know if you considered that, that's all. Thank you.
Yeah, thanks for the question, Chris. It is something, yeah, if I'm trying to make a nutrient budget of sorts, it would have to be, that would have to be included into that. But we don't have measurements for that. And I think you're absolutely right. Yeah, these, especially stormwater sources and just the runoff in the concrete and urban area is absolutely going to increase and change the contributions. It's something that should, for more comprehensive study, be looked at.
Chris, do you have recommendations on teasing apart the two different potential sources?
No, but I think it would have to be theoretical. I think it would be difficult to actually sample it, but it would be good. It would be nice to get a relative value from rainfall events. And if we could get into sampling, it would just add more clarity to what we believe. Thanks.
Awesome. Next question is to Jessica. Also as a comment, great job. What hourly precipitation did you use for your input in Model 3?
I used the NOAA National Weather Service data from Mānoa Valley. So can't remember the exact, numbers they've assigned that site. But I can follow up with more information if they want to know.
And then there's a follow-up. Are there other variables you sampled that can be incorporated into your models?
Yes, I tried to narrow it down today, but we have many different measurements, including other bacteria, not just Vulnificus, some fecal indicator bacteria and other pathogens. Yeah, I think we tried to do a pretty good job of getting the full sweep of water chemistry, dissolved organics, nutrients. We also have full community structure for microbial and eukaryote community identities.
I have sort of a follow-up to that. What can you give us like an estimate of your cost per sample, especially for Vibrio? You had mentioned that it is expensive to sample. Did you use labs, assuming at UH to sample? But is there like national labs that agencies like Department of Health can send their samples to, or just maybe expand upon that?
Yeah, so I did this analysis at Seymour Hawley, so the Center for Microbial Oceanography Research and Education at the University of Hawaii. So maybe Craig can step in with an approximate per sample. This is something that I was gonna throw together, but I don't actually know the answer. And I assume that there are government labs that they, like Department of Health, would run their samples at. And more sort of those probability of occurrence models, like presence absence sort of detection, rather than this full abundance measurement.
Well, then we'll move on to the next question. Can Model 3 take spatially distributed rainfall?
I don't know if anyone measures spatially distributed rainfall for such a small region. And the rainfall measurements that I'm using are in Mānoa Valley because the impact is not necessarily rain falling on the canal, but increasing the stream flowing into the canal, if that makes sense.
Tom, did you have anything to add with the rainfall atlas in terms of that spatial distribution?
Were you asking me, Michael?
Yes.
Yes, we have gridded rainfall products, and so they're designed to be used for these kinds of applications where you can translate them into put them into hydrological models. But right now we're working at a scale minimum time interval of daily. We're working towards sub-daily, but Yufan, who asked that question, is compiling data and implementing models at sub-daily timescales, like hourly. So I think that's a lot more relevant for a watershed like the Mānoa Pālolo watershed. So she may want to say something. Yufan, you want to add something?
Well, yeah, I just thinking because the discharge is not only from 1 point precipitation, since we have very high gradient precipitation. So you only use 1 point rainfall cannot really representative to the discharge. So if we can get like a spatial distributed rainfall, either daily we have now and hourly still on progress, then it will improve your model, I believe.
Yeah, I'd love to take a look at that.
Thank you.
Thank you.
Well, we are at the hour. I want to be respectful of everyone's time. Just thank you very much, Sean and Jessica, for presenting on your research.